Hello and welcome to Lecture 5 in the Inform Africa Toolathon series. In this session, we'll explore how to conduct advanced spatial analysis of location-based services (LBS) data using QGIS. This lecture is part of the Inform Africa project supported by the NIH-funded DSI Africa program, which connects researchers and data to improve health outcomes through data-driven science. I'm Dr. Xin Wu, a research scientist, joined by Dr. Chenfeng Xiong, assistant professor and principal investigator for Project 2 at Villanova University's Department of Civil and Environmental Engineering. Let's begin. Here's what we'll cover today. First, we'll learn how to load data into QGIS. Next, we'll explore how to join spatial and tabular data, such as linking shapefiles with population or trip records. Finally, we'll conduct advanced spatial analysis using LBS data to create meaningful map-based insights for mobility and public health studies. Let's begin by setting up our workspace. Navigate to the GitHub repository and download all files under the folder named QGIS module files. Then open QGIS and start a new project titled Joining Data QGIS Exercise. To add the Nigeria shapefile, open the Data Source Manager, select Vector, and locate the file named in Villanova GitHub mentioned in previous lecture. Click Add and you'll see the Nigeria boundary map appear in your QGIS project. Next, right-click the Nigeria layer and select Open Attribute Table. Here, you can explore the information associated with each state, including names, administrative codes, and postal codes. We'll use the field called admin1pcod as our unique identifier to join this shapefile with other datasets. This is an essential step because spatial joins require a field that exists in both the shapefile and the external table. Now we're ready to import the population data. Follow the same procedure you used for the shapefile, but this time choose delimited text instead of vector when adding a new layer. Select ADM Pop ADM1 2020 SV and add it to your project. Once it appears in the Layers panel, your workspace should now display both the Nigeria shapefile and the population dataset, ready for joining. Set the target layer as the Nigeria shapefile and the join layer as the population CSV. Select admin1pcod for both layers as the common field. Click OK, and now each state polygon in your shapefile includes population attributes from the CSV. You can verify this by reopening the attribute table and checking the newly added columns. At this point, it's worth asking which layer acts as the target layer and which one is the join layer, and which fields are being matched in the join process. Thinking through these questions helps reinforce the logic of spatial joins, a foundational skill in QGIS-based analysis. Now that we've successfully joined population data, let's practice joining another dataset, COVID-19 case data, for a single day. Here you can see the attribute table after the join. Each row represents a state, and the new columns on the right contain population and COVID-19 totals for that day. By viewing this combined table, we can analyze spatial variation in infection counts relative to population size, which is essential for understanding regional vulnerability. Next, let's visualize this joined data using graduated symbology. In the Layer Properties Symbology tab, choose Graduated and select the field containing the total population or COVID-19 numbers. Pick a suitable color ramp, for example, light to dark red, to represent increasing values. This classification allows us to easily identify areas with higher population or case counts, turning raw numbers into an intuitive spatial visualization. Here you can see the resulting map of Nigeria, colored by total population. Darker shades indicate states with larger populations, while lighter areas show less populated regions. Sometimes, it's more meaningful to map population density rather than raw totals, because density better captures how crowded a region is, which directly influences mobility and infection risks. Moving on to advanced analysis, we'll now bring in the trip number file from GitHub. This dataset represents daily mobility counts derived from LBS data. Add it as a delimited text layer, similar to how we imported the population CSV earlier. Once loaded, you can view columns like inner trip num, outer trip num, and geographic coordinates. This dataset lets us quantify mobility patterns across states, a valuable metric when analyzing behavioral responses to pandemic conditions. Sometimes we want to focus on a specific time period. Using QGIS's query builder, we can filter the trip data to show only trips recorded on a particular date, for example, June 6, 2020. This helps us analyze one day of mobility in detail, perhaps comparing it with COVID-19 trends or intervention measures. By isolating specific days, we can study how movement patterns change during key events or restrictions. After filtering, we can visualize trip intensity using graduated symbols or color scales. Each circle on the map represents a state, and its size or color corresponds to the number of trips or cases. This view allows us to compare mobility intensity across Nigeria at a glance, showing, for instance, that Lagos and Abuja have the highest trip volumes. Such visualizations help highlight where human movement is concentrated and where potential disease transmission routes may emerge. QGIS also provides a statistics panel for basic quantitative analysis. Here you can see descriptive statistics such as count, mean, standard deviation, and range for variables like trip numbers or COVID-19 cases. This quick summary helps confirm data consistency and provides insights into overall variation. For instance, identifying which states deviate strongly from the national average in terms of mobility. Next, let's examine the distinction between interstate and outer-state trips. In the Layer Properties, open the Diagrams tab and select Pie Chart. Assign inner trip num and outer trip num as the two attributes with distinct colors, for example, red for interstate and blue for outer-state travel. This setup allows us to visualize the proportion of local versus cross-border trips in each region. Here's the resulting visualization. Each state is represented by a pie chart. The red portions indicate within-state trips, while the blue slices show interstate travel. This map provides a clear, intuitive comparison. States with larger blue shares are more connected across borders, while red-dominant ones have more localized movement. Such spatial insights are vital for understanding how travel behavior contributes to regional spread dynamics. Thank you for watching this lecture on advanced analysis of LBS data using QGIS. If you have any questions or comments, we'd love to hear from you. Please visit our website at inform-africa.org and feel free to contact us by email. See you in the next session.